I'm Sam Barondes, and I'll be chairing this afternoon session. I'd like to start with just a personal brief reminiscence about my first meeting with Allison in 1992, and a brief explanation of how she came to UCSF. I was at that time the chair of the Department of Psychiatry, was eager to recruit psychiatrists, scientists, people who really knew psychiatry, loved psychiatry, but also were well educated in some aspect of neuroscience, and had by then successfully recruited 5 people, all of whom worked in cellular, molecular, and developmental neuroscience and genetics, things I understood and that were very close to my heart. But I was advised that maybe I should spread my vision a little bit and look at something else that I didn't understand too well. I think it might have been Zach Hall who suggested that. He was always prodding me to extend my horizons. And I heard about Allison. Since I go down to LA frequently because my kids live down there, I decided to go down and meet her. And I called her up on the phone, and she suggested that we have dinner together, and she suggested that we eat at a restaurant called Chinoise. I remember it very well. This was a trendy Wolfgang Puck restaurant at the time. In Santa Monica. I thought that was great. So I remember, I had never laid eyes on her, and I had seen her CV, and I remember going down there to Chinoise, and there was this beautiful young woman at the bar, and that was Allison Doupe, and so we sat at the bar for a little bit, and she just, you heard this morning the impression that Allison makes, especially if, she's on. And she was interested. She had already heard about the Keck Center and about UCSF, and she had finished her training at UCLA in psychiatry, did a full residency, and was in the midst of a very long and productive postdoc with Mark Kanishi. But she was ready to get a job, and this was sounding pretty interesting. So we talked, and I was very, very impressed. The big problem was that although we had a position, the Department of Psychiatry at that time, as in frequent years, had no space. I mean, we had expended the little bit of space that we could carve out of the former kitchen in the basement of Langley Porter and the former occupational therapy suite and things like that. But we were just finished. There was just no more place to go. So happily, the Keck Center was in the state of formation, and whereas we had a position which they didn't have, the Keck Center had space. And so a marriage was arranged. Allison had an appointment in psychiatry, an appointment in the Keck Center, and it seemed terrific. I must tell you that I had no idea what a jewel we had found. In my wildest dreams, I could not have imagined what a successful career Allison would have, what a huge impact she would have on UCSF overall, what a great role she played, not only as a scientist and as a teacher, but also as a leader who helped, amongst other things, build the Keck Center, now the Center for Integrative Neuroscience, build the Sandler building behind us, which was in a very touch-and-go mode, I know from personal experience, on a number of occasions, and she, along with Steve and others, played an enormous role in making that happen. So it's really amazing what an impact she's had on this community. And it's also amazing and hard to believe that she's no longer with us. So with that reminiscence, I'd like to introduce our first speaker of the afternoon, Russ Fernald, who is a good friend, a professor of biology at Stanford, someone who, like Allison, works on relatively simple systems and studies their ethological and physiological and mechanistic behavioral mechanisms, and a good friend of the Doupe Brainerd family. Russ.
Thank you, Sam. It's a real honor, of course, to be here and talking about Allison, or with you about my interactions with Allison. But first I'm going to tell you some of the science that we do and give you some idea of how we intersected. So all animals actually live in social groups, and the ones that do have a rank. And their ranking is established mostly by males through aggression. Giraffes, you may know, swing their heads which weigh around 400 pounds and hit the other animal in the solar plexus. It hurts, apparently. I haven't experienced this. And of course, it's maintained in other ways. They don't fight all the time. They maintain it through grooming or aggressive threats. And basically, this is because rank is so important. It allows you access to reducing conflicts, and it partitions resources. Dominant males get more food, mates, and territories. This is a picture from Darwin in 1856, who understood this well. Here's a dominant male and a non-dominant male. You can see from their postures, it's pretty easy to tell who is who in this case. So how is social status understood by the brain is the question we've been asking. How can we know that an animal recognizes they're a high status animal or not? So we needed a model animal that was actually had complex social interactions that were focused primarily on sex and food. This has a second goal, which is to attract undergraduates to our lab. And of course, because this reflects their interest, and many of our students actually became admirers of these fish, not just studiers of them. We have to be able to analyze lots of animals simultaneously because, of course, that's what matters. What's the social interactions? And we need to be able to measure things at all different levels: behavior, physiology, gene expression, et cetera. So I've chosen to work on this African cichlid fish called Hoplochromis bertoni, and it's not a fruit fly, it's not any of these other animals you heard about, even rats. It's actually a really wild living animal that we find really interesting because we can bring its whole world into the lab. So cichlids, first of all, fish are more than half of all vertebrates that are living. And the cichlid species are particularly interesting because they occupy these lakes in Africa. And recently the genomes of these animals, including ours, have been sequenced, so we now have genetic tools as well. So let me tell you, I'll talk today about this natural social life of the animal, the social status, how does it influence reproduction, what else is regulated by social status, what do these fish actually know, and I think this is sort of hard because people imagine that fish have a 3-second memory or something that's portrayed by the movies. It's not true. They actually are a lot smarter than that. And how is the social information transduced into cellular and molecular changes? How is it that social information gets into the brain and then changes things? So this is what they look like. So this is a dominant territorial male. He has this bright stripe, these spots I'll talk about in a minute. And this animal, well, it might be a female, but on the other hand, it might be a non-dominant male, because these two can interchange rapidly. They can change their phenotype. This guy can become non-dominant and the other one can become dominant. And that has manifest importance for their bodies and their brains. So where do they live? They live actually in Africa. I've already tipped that off. They live in Lake Tanganyika. And the early work was done by Max Pohl as the Europeans went and tried to see what they could steal from this part of Africa. They also sent scientists who were complicit in this theft. And Max Pohl was sent to ask, what can we take out of this lake? And he's shown here with his, not just his major research tool, his only research tool, the depth charge. So he actually went around the lake and blew up volumes of lake and took the animals out and put them in bottles and mailed them back to Belgium. This was the Belgian Congo at that time. And he basically wrote these large books about these fish and told us where they all were. There wasn't a lot of behavioral information. Let's just say he didn't concentrate on that aspect of their lives. He concluded they were all too small to eat, which was what's important to Belgium. But for us, it turned out to be a real gold mine. So they live actually in small shore pools. I'm here because of this. In the lake itself, there are crocodiles and underwater cobras, and it's quite a dangerous spot. Here, we could sit on the raft here, and although we spent all of our grant money bringing underwater diving equipment into the country, we actually sat on the shore and watched these animals as they lived in these pools. And these are stakes I've put in to mark out these points, these dark points here. And what you see happening is that all these circles are dominant males who've dug a pit in this detritus, this junk at the bottom, that they're true bottom feeders that eat on the junk. They dig a hole and they spend their time trying to find females. They have to spend on these females food. Their territory is about this big. They will swim into a school of animals, hope there's a female, try to lead her back and get her to spawn with them. So that's what they do. And the problem is a numerical one, which is there are about 1,200 animals, were 1,200 animals in this particular pool, only 600 males, 600 females, only about 60 or about 10% have territories. So you have, do the math, 540 males who don't have territories who need to eat. So the non-dominant males can easily pretend they're females, be led back to a territory, eat vigorously. The dominant male then says, okay, let's go into my grotto and spawn. And the male says, you know, I have other plans. And then they will go to other locations and actually do that repeatedly. And they're able to then survive quite well, but they're at the same time waiting for their chance to become dominant because these bright colored males who are the dominant males are the first ones to be eaten by the predators. The females in this social system, like every social system we know of, are completely in charge. They're regulating reproduction, so they can say, they swim into this area, they choose a male with a great territory, or like nice colors or nice moves, and then they can choose to spawn with him. And I'll show you a little bit what this is like. So this is a video which shows these animals in an unusual case of a dominant male right here who's doing pretty well, but he gets challenged by another male. So who swims in right here and will totally take this guy on. And you can see they're fighting, biting each other, hitting each other. And you'll notice other animals are watching them closely. And they're fighting, and now this intruder turns on his colors and begins to become dominant. And he's actually now gotten the upper fin. He's about to take over part of this guy's territory. And they decide wisely that instead of fighting all the time, they will just divide it right about here. And then from then on, this guy has a smaller territory, and he has one where he can start soliciting females. So the breeding in this animal is quite interesting. This is part of what appeals to undergraduates. The females actually brood the eggs in their mouths. So they lay their eggs and collect them immediately in their mouths, leading to a kind of an issue for fertilization. It's been solved because the male has these spots on his fin, and what he does after she lays her eggs, which you can see in this next video called Fish, Sex, and Videotape, actually shows the female laying her eggs, and she then picks them up. And this was cut for TV, so he doesn't usually go off and show off his colors, but you can see the egg spots here. But he'll now actually swim in front of her, put down his anal fin, put the sperm on his anal fin spots. She says, gosh, eggs I didn't pick up, collects them in her mouth, and fertilizes them. And he says, nudging her, let's spawn some more, and she says no. So basically, she then carries these eggs for 2 weeks, and when she opens her mouth, out swim small, functioning fish. These are really aggressive animals, so I thought I'd show this. This is a subdominant animal and a dominant animal, very aggressive in the number of behaviors per minute. Baboons, fruit flies, mice, zebrafish, none of them can hold a candle to it. Only this guy can be as aggressive, but he can't maintain it, which they can for hours. So they're extremely aggressive, so they have a lot of phenotypes that make them easy to study in the lab. So what do they do all day if they're not dominant? They mimic female behavior, and they avoid predators, but they turn out, we learned, that they watch other animals to see whether they can become dominant. So we have two socially regulated phenotypes, dominant males, non-dominant males. I've told you all these things. These guys are reproductively active, these aren't. And so we decided to pursue this and figure out what was going on, how this was regulated, because it's all under social control. So we raised small animals in either as dominant males or non-dominant males. And when we looked at their gonads, we found a pop quiz. These are non-dominant males and these are dominant males. Not only are they larger, but they're filled with mature sperm, and these are basically spermatogonia, precursors to sperm. So we then decided to look, as we all know, the brain controls the gonads. There are some exceptions, it turns out. And these are the outliers, trust me. All other vertebrates we know of use the hypothalamic pituitary gonadal axis. So other organs are not in control. So you have the brain controlling the pituitary, which has to release this peptide in a pulsatile fashion. I'll come back to that later. They then produce gonadotropins, which regulate the testes. And this, of course, is a feedback loop that's well known in all vertebrates. So we decided we'd look in the brain and discovered, using an antibody to GnRH, that the GnRH neurons in dominant males were very big, and those in non-dominant males were very small. And in fact, they were 8 times larger. The testes you've seen were larger and smaller. These had sperm, these had no sperm. These had high androgen, these had low androgens. So we had basically a phenotype of a social regulation of this reproductive axis. And we wondered what else did it change in the brain and in the behavior. So what else changes in these cells? First, we noticed that these cells change their dendritic arbors. So these are neurons, GnRH neurons, that are really very few dendrites in a non-dominant male, but a lot of dendrites in a dominant male. And these changes we suspected had something to do with the production of sperm when these animals became dominant. We looked then at slice cultures and showed that the dominant males had GnRH pulsatility that was correlated as opposed to the non-dominant ones. So now we're saying we think that these dominant males are able to produce pulsatile output of this set of neurons. And we really puzzled for this, how they could do this. And fortunately, a really incredibly gifted postdoctoral fellow, Scott Junty, who was trained at UCSF, made a transgenic animal. You're looking now at the base of the brain. These are those GnRH cells on one side and the other side, and this is where they project down to the pituitary. And what we showed was that you can record from these two of these cells, and they're all coupled with an unusual kind of connection, a gap junction. They have not normal synapses like you heard about this morning, but they're actually wired directly together. And when they're wired together, it's through these gap junctions, which we can block, and we know they're gap junctions. And basically, that's how they produce this pulsatile activity. They are able to actually generate pulses because they have these gap junctions connections. So we wondered whether maybe cortisol could be a part of the problem of regulating social status. So we looked at cortisol, and I'll say that Mimi Kao, who then went on to do a graduate work with Allison, was part of this project, looked at the cortisol levels. And indeed, when you put the animals together, for a while they're have equal amounts of cortisol, but then suddenly the non-dominant males, the cortisol levels go way up, the dominant levels, the cortisol levels go way down. So another interesting fact is that as these animals become dominant, they are turning off this high cortisol level, and these animals who are really laced with cortisol, which is the hormone that is responsible for stress responses, is actually pretty important. And of course, my colleague Robert Sapolsky said, well, these animals here should have liquid brains because they're getting so much stress, so much of this stress hormone. So we wondered about that, and we actually looked at the cortisol receptors. And this is what you just saw. Here's cortisol levels high in the non-dominant males and low in the dominant males. And we found that there are 3 cortisol subtypes, one of which is not very good. Here, cannot respond very well to cortisol. And this is work we did in collaboration with David Pierce at UCSF. And Wayne Corzan then showed that actually when these animals become dominant, they change the receptor subtype in their GnRH cells, put in this form rather than this form. So the switching forms at the receptor level. So thinking back, this is now all that we've changed is their social lives, and it's turning out to be a change in how they respond because they've altered the receptor subtype. So we then turned to some questions about the social life themselves. So we asked, actually this is another work from Stephanie White, who then went on to work in birdsong for reasons I don't still understand. But basically, she asked, well, can they be in denial about the status? This was actually seen by an undergraduate who noticed that when these animals became dominant, they did it really rapidly. When they became non-dominant, they actually took their time. And in fact, he witnessed them going into a shelter, turning on their colors, pretending they were dominant, and then coming out and turning them off again. So in the presence of a large male, they would not be able to be dominant in that large male's presence, but they could pretend they were by hiding from his vision and actually be dominant. So usually at this point, the women in the audience find this perfectly reasonable, and the men all ask, what? How could that be? So, but this turns out to be a really important feature of their lives. So it turns out that they have an attention hierarchy similar to that in primates. So this is a dominant male, and this is when he's in his shelter. And when he's not in his shelter, he will come out and attack other animals. Each of these bars is an attack. And this is actually a really interesting phenomenon. As soon as he comes out, he will go up and attack another animal. In this same tank are animals who are on their way to becoming dominant. They'd like to become dominant. We call them intermediate males. They are watching him very closely. And when he can't see them, they come out and turn on their colors, either coat a female or try to attack somebody. And when you can, they stop. So there's this kind of attention to his his watching them. So this kind of attention hierarchy is seen in preschool children, in lots of cases, faculty meetings. It's a very common kind of phenomenon where you watch what other people are looking at and you can tell actually what's going to happen. If you're not fully non-dominant, then you come out and whoever's available will actually attack you. So they were able to do this attention hierarchy, meaning they're attending to other animals. And this and other data suggested we had to look more closely at what they could actually know. So we did a test, which I thought was not going to work, and asked, can they actually tell who's going to win a fight just by watching the fight? So you can imagine the statistical problem that they're faced with. You're going to be a dominant male, well, you've got to go into a territory that has 60 males. You couldn't do pairwise fights with each of them. You must have to choose who you're going to fight with. So we did the experiment. In a natural condition, you'll have one male here who's wondering, can I beat one of these guys? So they actually did something, we studied something called transitive inference. You can do this when you're about 3 years old. If Mary is taller than Pete and Pete's taller than Fred, you know that Mary's taller than Fred because you can build a logical sequence in your mind from seeing pairwise comparisons. So we gave them pairwise comparisons. How did we do it? Here's a bystander. We have 5 animals, A, B, C, D, and E. We'll take them and move them into another compartment, and because we hold them for a second and increase their stress, they will always lose, so D will lose to E. So we let them be trained on A beats B, beats C, beats D, beats E, or the control is obviously A through E, all equally likely to win this fight. So the goal is to ask now, could this guy know, would B beat D? So he's never seen them fight. He's seen each of them pairwise fighting, but not individually in a row. So you test them. They're not very good with questionnaires, so we use something that we could reasonably rely on, which is all these fish and other fish, it's well known, will go and sit by the weaker of the 2 fish. So here they would always go and sit by E when A was shown, but even in a novel arena, they would always choose B over D. This is an undergraduate whose honors thesis was on the cover of Nature, pretty exciting for him. Now a graduate student at Stanford. He interviewed at UCSF, but you know, decided to stay. So here's transitive inference that they can actually do logic, simple logic, which is quite remarkable. Another kind of unsurprising result is that choosing losers makes females anxious. We began to think we needed to look more closely at the brain structures that underlie these social behaviors because we were finding more and more examples of how social information was getting into the brain. So we staged fights. We had a female. They'll only do this if they're gravid, meaning they're about to lay eggs. That's its big belly. So she'll see 2 males, and she'll choose one. She chooses them by going and sitting by that male's compartment. And then we'll stage a fight. And her winner, her chosen male will either win or lose this fight. So she's seeing 2 males, choosing one, and then randomly either that animal will win or lose. So she'll get information about this chosen male, and then of course we process her brain and we ask what was going on, what was she thinking of, so to speak. And basically we take punches of the key brain areas, and you don't really need to know much about that, but we then measure the mRNA expression of genes that map onto early action potentials called immediate early genes, and do PCR on those. So this is the brain regions that we looked at, and there's something called the social behavior network in all of us, and that is actually mapped out here. I could go through it in detail, but I think I'll just say that these are anxiety, spatial memory, reproduction, etc. So now what we do is ask what happened when she saw her animal win or lose. So here's the cerebellum. There's nothing about winning or losing. But if you look at when she's seen her chosen male win, she turns on her entire reproductive axis. These nuclei in the brain are all associated with reproduction, and she's upregulated the expression of this immediate early gene, meaning they're much more active because she has seen her male win. And when he loses, she turns on the lateral septum, which is a region of where anxiety is processed in all brains. So she's anxious when he loses, but really excited when he wins. We went on to ask what would happen if we did this again and asked them to make a choice after they'd see a male win or lose. And they basically almost never choose when their preferred male wins. So they always stick with the winner. Okay, so for male, it's another interesting feature, which is that fighting intensity depends on who's watching. So we have animals, a watcher and two guys fighting. They can't actually fight with each other because very quickly one would win. So we have a glass plate here, but we can actually watch them, the observer watch them. And no surprise, when they're being watched by a gravid female, they will fight harder. Very interesting. And when they're being watched by a much larger male, they will cut back on how hard they fight. So they're keying in on who's watching them to decide what they do. And they're doing this pretty actively. That is, they're doing it continuously in response to who's observing them. This, again, is something that's unsurprising to females, and males are always kind of puzzled by this result. Okay, so how is this turned into how's this information turned into molecular changes? This is a pretty hard question. We've been working on this for a while. I'll just tell you one example of what we've done. We put animals into this social group where they are basically a dominant male is bullying a non-dominant male, and there are groups on each side to make them feel like they're in a larger colony. We go in with an infrared goggles, and we perform what I call a bullyectomy. We remove the bully. So the next day at sunrise, this guy is here alone, and he wakes up saying, what happened? And you'll see what happens. Here is a video. This is work done by Sabrina Burmeister when she was a postdoc. So here is the dominant male. The non-dominant male is marked with this little white tag. And the dominant male is trying to lead this female back to this pot. She's not going, so he chases her, goes for another female, cleans up his pot. Maybe his pot was dirty. Now she's going to fight through the glass, leads again, and he's leading this female. She won't come, so he chases her away. These guys are, meanwhile, all looking out the window, hoping he won't notice they're still there. And basically, his full-time activity is trying to get one of these females to spawn with him and suppressing socially this non-dominant male. So now his moment of glory comes when he wakes up and, in fact, now he wakes up, and almost immediately you'll see he goes over and chews on the dominant male's pot. These animals are clearly in a much more different kind of constellation. He'll actually start digging and preparing this for soliciting a female. We know, of course, he has no gonads and can't spawn with them, but that doesn't stop him from trying to court them and bring them back to his pot. So he almost immediately turns on these colors and becomes a dominant-like male. And this dominance is really very important, obviously, for reproduction. So where to look for changes in neural activity? I'll just tell you a little bit about where we've been looking to see how this might happen. This is the fish brain, and this is the area that we discovered was actually activated. And we looked at EGR1, which is one of these immediate early genes. When an animal ascends, he turns this on. And when an animal, we looked at those, and they have either vasopressin or oxytocin. So both of them are implicated in other organisms to play a very important role in sociality. And so we basically looked at these in this fish and could show that both of them were important players. So here's actually AVP, arginine vasopressin, increased in ascending animals. Those are animals that are changing their social status from non-dominant to dominant. And similarly, we discovered that oxytocin, the same thing is true much more actively. So we now are in pursuit of these circuits. And we think of this in the following way, that this is just to say that GnRH is also upregulated, that basically there are two timescales. There's social ascent as non-dominant become dominant males, and that's in minutes. And then there's a longer phase. So we've now separated this into two different kinds of things. The social information comes in and goes to the GnRH axis, changes all kinds of things that you've seen, and ultimately results in reproduction. But the social information can come in much more rapidly and actually change these oxytocin and vasopressin cells. And we think that that's the circuit that's going to be the one that's responding to social information more directly. Okay, so I'll just say, I think I'll pass over this so I can talk a little bit about Allison. This is methylation and how it plays a role. I can talk to other people who are interested in this. But basically, status regulated by aggression and threat is really important. How does that effect get under the skin? And we think that animals and us are all collecting information all the time about others in the social system. you will walk into a room and look at people and say, are they male or female, or old or young, or rich or poor, and you can actually use that information or not, but it guides what you're going to do. Animals do that too, and these animals, we've just shown a few cases of where that happens. And these dynamic changes are really important in changing the brain in circuits and receptors directly as they learn what is going on. And there are at least two areas that go through the preoptic area. So let me say these are the people who did the work, Carolina Hu and Scott Junti, the one who did the great transgenic animals at the Marine Aquarium. So I'd like to turn now and say a few words about my interactions with Allison. I met Allison in 1995, introduced by Darcy Kelly at this neuroethology meeting, and it was really a wonderful time. We had a great set of discussions, and after that, communicated by, at that time there were telephones connected to wires, so we talked to one another and had a really great set of conversations. And then a couple of years later, at the 1999 Neuroethology Gordon Conference, I was talking to a friend of mine, an Englishman, and he said to him, what do you think we should do? And he said, why don't you go punting on the cam? So I said, great. And Allison and Darcy and Darcy's longtime collaborator, Martha Tobias, went, and we went punting on the cam. And you may wonder why Allison is finding everything so hilarious. She was laughing, and actually, I was providing full-time entertainment because I was trying to use this pole, and although the guy had told me, be careful, don't get it stuck in the mud, or you'll end up in, I was getting it stuck in the mud, I was hitting the trees, I was actually soaked as I was doing this, you could see. And Allison was in her usual case saying, you can do better than that, come on, get out, don't do this. And I just remember vividly this great way she had of teasing me. And so I said, okay, you do it. And so she did, and of course she could do it with her eyes closed, never got wet or anything. So she was perfectly capable of this, and then teased me about that as well. So it was really a wonderful event. Then sometime later, we hit upon the idea of having, we were actually toasted. I didn't mention that alcohol may have played a role in this. But we actually got a free a glass of warm beer for not having fallen in, which most Americans do, apparently. So then sometimes later, we decided we would try to have joint lab meetings. And Mimi Kao, who was then the undergraduate who worked in my lab, sort of motivated this. So this was birdsong and fish, and how could we interact? At first, it was a little bit like two partners, one dancing the tango and one dancing the rumba, not quite knowing how to talk. But we had wonderful times, and I think some of Allison's lab members who are here remember these times, always culminated in a great meal where we actually ate very well and had a good time. And most of them resulted in really interesting discussions, and I think I was guided by her pursuit of the circuitry to go more deeply into this field study that I had done. And we had sort of occasions of wearing funny hats and so forth. And then our lives jumped ahead, and Allison and Michael had these wonderful two boys. At about the same time, our daughter Leah and her husband Guy had their daughter, Mirabelle, our oldest grandchild. This is my wife, and this is Allison and the boys. And here is Mirabelle at the second birthday. So we introduced Allison and Michael to Guy and Leah, and here they are at the second birthday. This is Mirabelle pushing the twins on this until one fell off, and then she just kept pushing. She's now 10, as they are. But they became fast friends, and it was really wonderful that we had this connection with them because we got to go to lots of family events that involved lots of food and small kids and conversations with Michael and Allison. Our role was to produce puppet plays, including one we did on Easter. Some of you may have been there. Some of Allison's family came. So this was a case in which the Easter Bunny in this puppet show was mugged and lost all his eggs. And fortunately, a Canadian Mountie was around to rescue the situation. So I had specially acquired a Canadian Mountie to perform in this case. And Allison and Michael were always very generous in their interpretation of our puppet shows. We had a long series that we did at Leah and Guy's house where Michael Ellis would just say, keep it up. So, writing a play that appeals to people from diapers to 60, not easy, but we had a pretty good time, and it was partly because the two of them were really encouraging. So I think the nice thing about this celebration is, of course, we all loved and admired Allison, and she's leaving behind not just all these wonderful students in her image, but these two boys who carry so much of that great soul that she has. That's it. Thank you.